Hi all, I'm Poorni, working as an assistant professor in the Department of Biotechnology. I'm handling the subject called Bioprocess Engineering, and in today's session, I'm going to discuss about the mixed culture fermentation. So when we talk about a fermentation, it is said to be the heart of any bioprocess unit. So when we talk about a bioprocess unit of a fermentation, it is said to be the conversion of a substrate into the product with help of a activity of a microorganism. So with the action of a microorganism, the substrate is getting converted into the product. And when it is converting into the product will be of a desired one, undesired one, and it also said to be the byproducts that can be formed. So that can be purified with the help of your various downstream processing technique. That is a separate topic that has to be discussed in a later phase. So when we talk about a fermentation, we might be knowing that there will be a single usage of an organism will be utilized for the substrate for the conversion into the product. So in today's topic, I'm going to discuss about the mixed culture fermentation, which means that this is said to be the fermentation where there are two types of an organism or more than two types of organisms will be utilized for the conversion of the substrate into the product. So this type of an organism, it might be of a known species or it might be of an unknown species also. So when we talk about the mixed culture fermentation, this inoculant might consist of a two or more organisms that will utilize the substrate as an input what we are going to give as a raw material that is being converted into the product. So when we talk about a mixed culture, it might consist of a bacteria or it might be consist of a fungi or it might be of a fungi and a bacteria also. So in the bacteria, if we see, we'll be adding a two types of a bacteria, or if you're fungi, there may be a two source of a fungi can be added, or it can be added in a different combination of bacteria and a fungi, or fungi and a yeast also can be added in which where your mixed culture fermentation will be taken place for the product formation. So when we talk about your organism of a mixed culture, that has to be stored in a lyophilized condition, that has to be stored in a liquid nitrogen cylinder for almost of a temperature of about minus 80 degrees centigrade. So here the main problem in a mixed culture fermentation is said to be the storage and the maintenance of a culture. Once we are mixing up the two cultures, if we are using for the fermentation process, that has to be stored in such a way to prevent their contamination by storing it in a liquid nitrogen which is below the minus 80 degrees centigrade. So here when we are going to do the fermentation process, we need to remove the culture from the stored and preserved where it has been stored in the case of your liquid nitrogen. Then it has to be prepared in a fresh inoculum for the fermentation process, or we can tell it has to be inoculated in the production medium for a fermentation process to be carried out to form a particular product. So the next one, when we are using this, when we will be using this type of a mixed culture of a fermentation. So here we will be utilized for a human purposes to produce a food, as the demand has been increased nowadays by the human population. In order to cope up their demands, we are going to produce the particular product which has been utilized by the human in a large manner. That time we'll be using a fermentation process called a mixed culture fermentation by the combination of the two organisms for a particular substrate to give a product. So here the next one we'll see why we are doing this. This is doing for our industrial biotechnology to produce a high amount of a product in a large quantity, as well as the product should be in 100% efficiency for a human consumption. That is, when before entering into the market, it has to be entered into the market as a commercialized purpose that can be utilized for the human for our well-being. So here, when we talk about a mixed culture fermentation, the complexity is said to be the substrate consumption in a nature that is being utilized by the microorganism. So one source of a microorganisms might use a carbon as a major nutrient. The other source of a microorganism might use a nitrogen as a major source for them. So here the complexity that has to be broken down in the case of a substrate consumptions for a product formation. So when we talk about a mixer, this we'll be using for our conversion of a substrate into the product using a pure microbial culture for the product formation. So how this can be taken place? So here when we talk about your substrate that has been converted into the product, how it has been involved. So here in this picture we can see when we having a substrate that has been acting on a particular microorganism, so that this microorganisms utilize the substrate, so it's started to getting converted into the product. So once it has been converted into the product, this further it will be acting on other organisms, so that your conformation change will be occurring, so that the product will be increased in a high amount or in a high efficiency we will be getting. If not, when we talk about a microbial culture of a mixed one, this is said to be the direct formation. If this is of an indirect one, it will be dependent. So here, whatever the product has been formed, that has been from the substrate, that has to be reacted with the another organisms at a later time. But here in this case of this picture, the substrate is getting into the product by the reactions with the microorganism first, then it will be a further confirmation of your substrate that has been acted with the microorganism too, so that it is getting converted into the product. So this can be of either a direct microbial interaction, or it might be the metabolic dependent interaction. So here when we talk about the substrate which will be converting into the product with the help of a microorganism one, so that it's getting converted into the product. If not, the substrate will be acted on a microorganism one, that will be converted into the product. Then this product, which means that it is said to be the undesired one, which will be acting on a other organism to produce a desired product. So this is said to be the metabolic dependent interaction, which means that the byproduct also which should be acting on a particular microorganism, so that your further product will be formed. So when you talk about your mixed culture of a fermentation, this can be used for a various products like your biogas preparation or a bioethanol production, or your biofuel generation, so etc. we can talk about. So this is not a developing one, it's not a developed, still it is in a developing stage. And since we are using in a mixed culture of a fermentation, so what organism which plays a major role in the case of a production of a product is being a unquestionable one, an answerable question. So here in this case, when you talk about your first example, we'll see about the biomethane gas production. So here how this biomethane gas production is being taken place. So here this is said to be the reactor where you are going to give the input as an influent, in which there will be a mixture of an organism will be acting on it. That which is your influent, whatever we are going to give, that is said to be having a microorganism as a mixed culture, which when the reaction is being carried out, this is in the conversion of your biogas. So here how these reactions will be occurring. So once the biogas has been formed, it will be taken to the effluent. So how this reaction is occurring? So this reaction is occurring in a step-by-step manner for the biogas production. So in the first step, whatever the source we are going to give, that will be utilized by the organism, either it might be M1 or M2 organism. So here these two sources can be utilized by the microorganism, either the carbon as well as sugar and amino acid as a nitrogen source, to produce a product as a CO2 and a H2. Then it will be converted into the acetate. Then further this acetate will be converted to your reaction with the help of a microorganism, that will be converted into the methane. So whatever the organism as an influent which is containing the mixed culture of a fermentation is being carried in a fermenter to produce a biogas, along with the byproduct as the carbon dioxide and hydrogen evolution as well as the acetate. production also will be there. So this byproducts, whatever it has been formed, that can be converted into the final form of a biogas in the form of a methane. So here, when we talk about this one, this is said to be the metabolic dependent products. So it is each and every step, whatever the product as an undesired one is there, that is dependent on the another one to form a methane, in which it is not said to be the direct dependent of your reaction. So the next one we'll see about how this mixed culture fermentation is useful in the case of a industrial wastewater treatment. So here in this case of industrial wastewater treatment, it is said to be where we are going to give the effluent, which means the water as an coming out as a waste, that has to be treated with the help of your mixed culture. So that can be provided with the help of your aeration tank, and where the reaction is going to be taken out, that is said to be the secondary effluent that can be regenerated as a waste one. So here in this case, how the reactions will be taken place. So whatever the effect we are going to give, that will be passed to the aeration tank, in which the air is being provided with the help of an aerator for the reactions to be carried out, in which this mixed culture, which contains your various components in the case of your cell wall, that will be reacted with your mixed culture, so that it will, the water will, whatever the components which are present in the water, that will be reacted with the cells structure, whatever the components they do have. So it will be converted the industrial wastewater into the recyclable water for our some other purpose in the case of the industry, like a watering purposes for the gardens. So here, the whatever the waste that has been settled, that has to be clarified or cleared with the help of your clarifier. It can be recycled back to the effluent for a further, again, regeneration of a process. So the next one we'll see about a bioethanol production. So when we talk about the bioethanol production, so this is said to be a production of a ethanol with the help of a microorganism. So as we are aware that there are n number of products that can be produced with a single specific microorganisms by utilizing the substrate. Right? But in the case of mixed culture fermentation, we are going to use the microorganisms in a mixed condition. So there will be a gram-positive bacteria with other source like a yeast, or gram-negative bacteria with other sources. The yeast can be used for our mixed culture inocula for a bioethanol production. So as we all know that there are many ethanol can be produced in a chemical way. Okay, not a bioethanol, it's a chemical method of a preparation of ethanol that can be produced in the case by taking the source from the industrial source, whatever it may be taken. That is the industry, what they do take by the from the petrochemical industry, whatever the effluent it has been there, or a petrochemical ethylene that can be used for ethanol production by the chemical means. But in the case of plant source, if we are using, so that can be used as a sugar cane or a corn or a wheat and a sugar beet, so that the fermentation can be carried out to produce a ethanol. So we call that ethanol as a bioethanol under the sterile condition that has been produced. So as we all aware that there are a gram positive and a gram negative bacteria can be used for our ethanol fermentation process to produce a bioethanol. So here, this this production of a bioethanol from starch, if you are using, it needs an alpha amylase enzyme. Since amylase enzyme is said to be the costliest one, costly, so that the enzymatic sacrification for converting the sugar into ethanol can be taken place. So similarly, the sacrification and fermentation can be done with the mixed culture, which reduces the cost. So instead of using your one specific organism with the help of an enzymatic activity for the conversion of the sugar into the alcohol, we can use the same thing with the help of a mixed culture, which will be reducing the cost of the process. And then it can be used for a transportation purposes. So the next one, there are various feed sources can be used as a source for a conversion of your biomass stock into your energy form. So here, what are the various factors that influences? So what are various factors that influences are a lignin content and the crystallinity of the cellulose of a particle size, the digestibility of the cellulose, and a hemicellulose in the lignocellulosic biomass. So that also has an impact on a bioethanol production. So here we have to since this is said to be the factors affecting the bioethanol production, this can be redone with the help of your using the carbon source, which is having a 6 carbon and a 5 carbon sugars to complete conversions by the pure culture of a mixed fermentation. So the next one you'll see about there are a genetically engineered microorganism also can be utilized to produce the same bioethanol production. But using the genetically modification of an organism, it has a higher effect, but it has a high efficiency that can be used as a fuel purposes. But still it is said to be the costly and cost effective one and high labor intensive one, as we need with the help of a genetically modified organism. This is said to be a good efficient one, but decrease in utilization of the equipment and the labor intensive. So here this can be studied with the help. So this problem can be overcome by studying or using the genetically modified organism that can be derived from the lignocellulosic biomass base that and a mixed culture can be used to produce the bioethanol. So here, these are the two examples where it has been utilized as a mixed culture. So what culture they have taken? The one is the yeast and the another is the Fusarium species. The next one is Zymomonas mobilis along with the Candida tropicalis of some mixed culture for ethanol production. So here it has produced and compared with the Saccharomyces cerevisiae and Zymomonas mobilis. Zymomonas mobilis has produced almost 98% of a bioethanol. So this bioethanol can be produced as well as a saccharification, liquefaction, as well as with the help of an enzymatic digestion also. So this has an efficiency of a 98%. So this prominently clears that the mixed culture for industrial biotechnology can be utilized for a bioethanol production. And then we'll see about a biofuel production. So here we talk about a biofuel. So as we all know that there are n number of renewable source of an energy can be used for a fuel for a energy purpose. So here we are going to utilize the organism as a mixed culture for your biofuel production. So how it can be done? So because it has a potential to replace a significant portion of fossil fuels, which will be affecting our food supplies, as well as it gives a numerous hazardous environmental impact of our human. So here, in order to overcome that, we are using the mixed culture of a fermentation process to produce a biofuel. So in turn, which will not affect our environment, so that it does not affect our human health. So here, this is the thing how the organism has been involved in the production of an electricity. So when we talk about this one, here we have the anode region in which we are going to make the waste, which consists of n number of organisms. We cannot predict which organism is there in the waste. We can characterize what are the various organisms by the serial dilution method to isolate a peculiar microorganism. But here, as we are going to give as a base to for your as a source for your energy production in the form of a fuel. So here the example is said to be the microbial fuel cell. So we call the electricity production by the microorganism is said to be the microbial fuel cell that has to be placed near the anode region. So when what will be acting as the cathode region in the sense, here it is said to be the buffer. That is a buffer which will be containing a salt buffer solution. So when we are connected to the current, connected to the multimeter, we can measure there will be a movement of electrons or ions that can be moved from the anode to the cathode region. or cathode to the anode region. So there will be a connectivity. This connectivity we call it as a this called as a salt bridge, where your exchange of the ions taken place between the anode and the cathode, or a cathode to the anode. So here in this case, it will be producing, when there is an exchange of an ion occurs, there will be an intimation on a multimeter on a how much amount of a electricity has been generated with the help of this base, since it contains a mixed culture of a microorganism. So in which way the microbial fuel cell can be used as a microbial consortia to produce electricity, which brings a large number of attention nowadays. So a lot of work is being carried out by varying the source of a waste that can be collected from the different source. So here this can be categorized into two categories. The first one is a mediator, the second one is a mediatorless. So when we talk about a mediator, this will be acting as again the substrate, so that your organisms which have been present in the base can be motivated, or or it will be encourage the organisms to produce a large number of your fuel, electricity in the form of a fuel. Can be used as a fuel. And when you talk about a mediatorless, this will not be adding, like your enzyme if you are adding and the enzyme if you are not adding. So here this mediatorless is said to be without the addition of your substrate to the feed, how this organism has utilized the fuel so that is getting converted into the fuel form. That can be done with the help of your mediator. So there will be a comparative study, and this makes the modification of your various pathways to advanced study of molecular tools to produce electricity. The next one is a biohydrogen production and a butanol production. So hence here we can see how there are two stages in which your methane biogas can be produced. The first stage is there will be a first mixture of a culture. That is the first culture which will be having, where you are going to give the base so that it will be treated to give you the hydrogen, as well as there will be evolution of a carbon dioxide also. The second stage, there will be a second stage of an organism which will be utilized in the case of a mixed culture, that mainly of a methogenic bacteria. So it gives you the action on it, it gives you the methane. And when we talk about a butanol production, here in this case of a butanol production, there will be an organism which uses called a acetogenesis process and a solventogenesis process. So here we use an organism as an acetogenetic bacteria. So acetogenetic bacteria. So here in this case, we are going to take the lignocellulosic base as a source for the production of a butanol. So here we are going to give your first treat a lignocellulosic base, then it will be treated with your mixed culture by adding your whatever the substrate which we needed for the microorganisms, that can be treated so that it can be converted into the butanol, and that butanol we can derive, and the remaining thing it will be recycled back as an effluent for a further treatment of a process. So when we talk about the dialysis, this is a picture for a dialysis two-chamber bioreactor system that is used to study how these organisms which are present on a first chamber is getting reacted with the second chamber. So there is a mixture of a culture which will be present in that can be poured, and this membrane will be having a porous one, so that the movement of organism which will be occurred in this random manner, so it will be used to study how this interaction is being taken place. By without a mixing, we are going to fill the culture in a one compartment and in the next chamber, so that how the purification is being taken place can be studied with this dialysis two chamber bioreactor system. So when we talk about your challenges, so here, because even though when we are using a single microorganisms as a source for a product conversion by utilizing the substrate, here we are using a mixed one. So it is not an easy technique to determine the population size of a cell. So as we are using a two source of an organism of a known or an unknown species, or a different species, the population size cannot be determined. So that is a challenge that can be. Then how the interrelationship is being occurred between these two culture. That is that two organisms if you do, which is having a high impact on the production of a particular product by utilizing the substrate, that analysis has to be done. And then the genetically modified organism, even though it gives a engineered microorganism for a bioethanol production, but it has an efficiency of a high 100%, but still it cannot able to cope up with, because it is of a cost, labor cost, that is a very high cost, and it needs a lot of a genetically modified organism that has to be done by altering the genes for the production. And when we talk about the next challenge, is it will be the fermentation contamination. So even if we are using a single source of an organism for a substrate conversion into the product, here we are using a two or more than a two type of an organisms as a mixed culture. So that fine, surely it will be lead to the contamination. That is it to be the next challenge. So as we are mixing of a culture, here it means that the consistency. So how the consistency has to be, the consistency of the inoculum should be not in a turbid manner. It has to be in a somewhat liquid manner. So since we are using a two culture as a mixture in the fermentation, so the consistency of a fermentation inoculum or a medium will be of a more turbid, so that it impacts your product formation. So here when we talk about the mixed culture fermentation process, this represents your necessary enzymatic conversion in the reactor for engineering the microbial consortia for the production of any form, rather than using the synthetic engineer consortia. This can be used as a model system for studying your molecular tools that has been developed, so that the bioaugmentations can be able to produce additional fermentation, so that can be produced the product or enhance your existing bioprocess. So always this industrial biotechnology is a promising department in which your organisms can be used rather than in a one, and it can be used as a mixed culture for a production format by the conversion into the substrate, the conversion of the substrate. So this makes your interdisciplinary approaches, since it makes your genetically modified organism, everything that has to be taken in a molecular level, and finally it is taken to the process level for production of a product by utilization of the substrate by genetically modified organisms. It is said to be in a interdisciplinary approaches. So when we talk about the advantage, what is the major advantage? So the advantage, so why we are going for a mixed culture? Because we do need a product in a higher amount. So the product yield is a higher. So we are going, that's the first advantage. Then since we are using the two types of an organism for the mixed culture fermentation, so the growth rate should be a higher one. So that is a second advantage. The next one, multi-step transformation. So here when we are going to the multi-step transformation, it indicates that the substrate is converted into the product, and this substrate, when it is converted into the product, there are n number of steps which will be in. It will not be directly converted into the product from the substrate, which means that there are n number of intermediates and the byproducts and whatever the undesired products which will be formed as an intermediate. The unstable product, as we all know, that has to be converted into the product. So there are a multi-step transformation process which will be occurring, which means that there will be a change in the shape of a microorganism due to the activity of a substrate on a particular microorganism. Then the next advantage is an exclusion of unwanted microbes. That means the contamination, it has to be avoided. We have to maintain in such a way, we should maintain the culture as a subculture rather than disturbing the mother culture. So we have to keep on inoculating once in a 10 days for maintaining the aseptic condition or a sterility or a pure condition of a culture without the contamination. The next is a strain selection. So we need to select the strain in a proper way so that it will be avoiding the toxic product formation, so that it will not involve in a product formation inhibition. The next is that it has to give a more protection against the contamination. As we all know, the contamination has to be avoided even if we are using a single source of a microorganism for a substrate conversion into the product. The same way, we have to take a more care against a contamination for a mixed culture. And it is easy to maintain a mixed culture medium indefinitely. It is a big task, but if we are maintaining it in a periodic form, it is used to carry out a successful product formation in a repeated way. So that gives you the product in a higher quantity with a 100% efficiency. So when we are using this mixed culture, we have to make use the cheap and the pure form of a substrate that has to be used. So here the next advantage is it requires your optimal factors for your product formation. So it requires the optimal growth factors to achieve the growth rate by providing the necessary nutrients in an optimal concentration. So if the organism is needed to the carbon source, that has to be given in a maximum optimum level. We should not give the carbon source beyond the optimal level or below the optimal level. So it has to be in optimal level. We have to make sure for a mixed culture fermentation to give the optimal performance. And the last advantage is it to be, we need to stick on to the symbiotic relationship, which means that if we are using the bacteria and the yeast, there should be of a relationship that has to be existing. So one should not take over the other microorganism and should not dominate or should not suppress the activity of the other organisms on the case of a mixed culture fermentation. So the next we'll see about the disadvantage. So when we talk about the disadvantage, as we all know that even if you are using a single source of a microorganisms for the substrate conversion into the product, here we are using a mixed culture of a different source of a microorganism. So here it is difficult to study how this organism is utilizing the substrate if we are having more than one organism is involved. So how the fermentation process is carrying out, whether the microorganism one is acting on a substrate to give the product, or M2 is microorganism second one is acting on a particular substrate to the product formation. So that is the first disadvantage. So difficulty in studying the fermentation kinetics of a product formation by the substrate utilization. The next one is the product and the microorganisms is said to be whatever the things we are going to make as a product, we are going to do it as a patent form. So we are going to file the patent by the patentee for the patent right of an information. That becomes a more difficult one by defining the product and the microorganisms relationship. So that means you're one of the disadvantage while filing the patent. The next one, as we all know, the contamination is the main source of a undesired product to be formed in a large amount rather than the desired product. So it is more difficult to detect and control the contamination. And the fourth one is when we are using a 2 or a 3 culture orbits together, it requires a more time and a space for the growth, for the development by taking the time for their adaptation, for the utilization by the substrate of a particular microorganisms to be converted into the product rather than the one source of a culture if we are using. So it needs a more space and a time for producing the inoculum so that it will be impacting on a product formation. So here, as we are using the microorganism of a mixed culture, it is and be able to, the disadvantage is controlling of the optimum balance among the microorganisms is one of the very difficult tasks. So if we are selecting the acidic condition of a bacteria, we have to both the source of a bacteria and a fungi should be in an acidic condition and not to be in a basic condition. So if we are knowing that if it has been since if there is a pH fluctuation, the difficulty in controlling the pH level is said to be of a more difficult one to obtain or control the optimum balance among the microorganisms. So these are the various disadvantages of your mixed culture fermentation in the bioprocess. Even though it has a high efficiency to produce a product in a larger quantity, but still it decreases the cost manner, but it has a disadvantage of your contamination, as well as balancing the optimum level for the organism, and which organism is giving a particular product by the interrelationship between the microorganism and the substrate are very difficult to predict.